Hello dear students, in this video we are going to discuss antigen antibody reactions. Since we have already seen what is antigen, what is antibody, so today we are going to see what happens when they react with each other. So in this class we'll be discussing the following topics, that is what is the definition of antigen antibody reactions, different type of reactions, what are the various types, most important precipitation, agglutination, ELISA, and immunofluorescence. So these four are the most important antigen antibody reactions in exam point of view. So that we'll be discussing in detail. So a previous year question which was asked from this topic, one is ELISA and the other one is agglutination. And the other probable questions are precipitation and immunofluorescence test. So regarding a definition of antigen antibody reaction, so we have seen an antigen class on antigen and antibody. I've told you antigen or antibody, each of them should react with each other in an observable manner. That was the definition. No? So if you are allowing antigen and antibody to interact with each other during an immune reaction, they interact with each other in a specific manner. That specific reaction or interaction is what is called as antigen antibody reaction. So in this picture, you can see an antigen with four different types of epitopes, and each type of epitope has stimulated the production of a kind of antibodies specific for that epitope. So we get four different type of antibodies for this antigen, and when these antibodies interact with this antigen, most probably you'll be able to visualize it in a particular manner, like in the form of a precipitate or in the form of clumps, or you can make it visible by certain other methods. So these are all antigen antibody interactions. Why we need to learn about antigen antibody reactions? The importance. The main thing is they form the basis of immunity or protection against various infections. So we know when an antibody is produced, it will bind to a virus, it can neutralize the virus. An antibody binding to a bacterium, it can trigger phagocytosis, it can induce inflammation, it can activate complement leading to cell lysis. So everything which gives protection against infections, all these things are linked to antibody and the reaction between antigen and antibody. And we have certain specific type of antigen antibody reactions like agglutination, precipitation. Those things are important in diagnosing disease. Suppose if we want to detect the presence of antigen in patient's blood, for example, hepatitis B, if you want to diagnose, you are going to find out whether the hepatitis B surface antigen is present in the patient blood. Then you are going to use monoclonal antibodies against hepatitis B antigen as a reagent. Similarly, if you want to find out HIV antibodies, whether it's present in patient blood, you're going to use the HIV antigens as the reagent. So we are going to visualize the antigen antibody reaction, and thus we are trying to find the presence of a particular thing in the patient's blood. So this is used in diagnosis of various infections and other, not only infections, other diseases also like malignancies or any other kind of immunological disorders. Not only that, antigen antibody reaction can trigger hypersensitivity reactions. So hypersensitivity is mediated by antibody. Autoimmunity, in certain situation, body produce its own antibodies against its own antigens. That's called as autoantibodies. So reaction between autoantibody and self-antigens can lead to autoimmune diseases. So everything is related to antigen-antibody reaction. So that is why we are going to learn about it. So we'll see what are the different types of antigen antibody reactions. The most important ones, that is important in exam point of view, I have put it in bold: precipitation, agglutination, and enzyme-linked immunosorbent assay, which is called as ELISA. All others, complement fixation, neutralization test is also important. In mostly in like the complement fixation test, we are not doing that nowadays. It was a previous older test, older immunological test. Then immunofluorescence and radioimmunoassay has also got a wide application in the diagnostic field. So all these are the important types of antigen antibody reactions. So first of all, you're going to see precipitation. So what is precipitation? It's an antigen antibody reaction. And what is the definition? See, when an antigen is soluble antigen, if it's a soluble antigen, when it reacts with its antibody in the presence of electrolytes like sodium chloride at optimum temperature and optimum pH, there is a complex formation, and this complex will be in the form of an insoluble precipitate. So you have an antigen which is soluble and you have its antibody. When these two combine together, it will form a complex which is in the form of a precipitate. So this is called as precipitation reaction. The most important part of this definition, Any antigen antibody reaction, when you define, all these things will be there. Antigen reacting with the antibody in the presence of appropriate temperature, pH and electrolytes, leading to formation of a complex. So what kind of complex it forms is different depending upon the type of antigen. If your antigen is soluble, the complex will be very small in the form of a precipitate. If the antigen is insoluble or particulate antigen, particle-like antigen, it will form a larger complex in the form of agglutination. So that is the difference. So in precipitation, you're getting insoluble precipitate. It's very smaller complex. And usually, if you have a reaction taking place in a tube like this, antigen antibody reaction takes place, there is this complex formation or precipitate formation which gets settled at the bottom of the tube. That's what normally happens. You have some modified forms of precipitation reaction like flocculation. What is flocculation is, instead of getting precipitated, if suppose the complex is getting suspended in fluid as floccules, the suspended form of the complex is called as floccules. If it gets suspended, you call it as flocculation test. The most important type of precipitation reactions are flocculation. And the one which we do even now, we use that as a very important screening test for diagnosis of syphilis. So for diagnosis of syphilis, we have two tests, VDRL and RPR test. These two are actually a kind of precipitation test which you can call it as flocculation because the precipitate gets suspended as floccules in the fluid. So in this is you can see a slide here, or you can call it as a tile with depressions. So VDRL test is done in this. So you put the antibody, you put the antigens, and you add the patient's serum. Suppose the serum contains antibody against these antigens, a precipitation or flocculation occurs, and you can visualize the floccules under microscope. So that is how you do VDRL test. And this is a modified kind of VDRL test that's called RPR, Rapid Plasma Reagent Test. Here in this figure, you can see in 1 and 2, there is no precipitate formed. Whereas in 3 and 4, you can see this fine kind of tiny black kind of precipitates that is actually positive. So this is RPR positive or reactive. 3 and 4 are reactive, 1 and 2 are non-reactive. So that is a flocculation test. Now, how this precipitation reaction occurs, you have to know the mechanism. And a theory has been proposed, a theory of lattice hypothesis. It's a hypothesis, that is lattice hypothesis. And what this hypothesis say is, suppose you're taking equal amount of antibody in all the tubes. And if you're increasing the amount of antigen that you add to different tubes, what you can see is initially there is no precipitate formation. Why? Because in this figure, you can see there is excess of antibody in the tubes and antigen is in very small concentration. So since there is excess antibody and very less antigen, no precipitate is formed. And in this, when you increase the amount of antigen, what happens is at a particular point, antigen and antibody will be of equal concentration. So you call that a zone of equivalence. When antigen and antibody are of equivalent proportion or equal concentration, you will get the best precipitate like this. So here you can see there is a precipitate formation that is called lattice formation. So the lattice formation is the best when antigen and antibody are of equivalent proportion. Again, when you increase the amount of antigen, antigen becomes excess and antibody is low, you don't get a precipitate. So you don't get a precipitate in the first set of tubes and the last set of tubes due to antibody excess and last due to antigen excess. In between you get good precipitate because there is equal amount of antigen and antibody. So when you get this precipitate in the zone of equivalence, that is the lattice formation, that's called the lattice hypothesis. And why you don't get it in the higher antibody and higher antigen concentration, you call it as a prozone phenomenon. When there is excess antigen or when there is excess antibody, there is no precipitate formation. So that is the mechanism. Now, what are the different types of precipitation reaction? I told you most of the time when you simply take liquids in a tube and try to do this antigen antibody reaction, the precipitate gets settled at the bottom of the tube. So instead, sometimes if you take antibodies in a tube and you add antigens, soluble antigens, sometimes you can get a ring formation at the middle of the tube. That's called a ring precipitation. So this is the place where there is a zone of equivalence. They move, antigens move from above, antibodies move in between, they meet. At a zone of equivalence, you get the precipitate formation. So in this tube, you can see that ring formation. So ring precipitation is a common kind of precipitation reaction. Other than ring, the one which we have already learned, flocculation test, that's another type of precipitation. Then if you do precipitation reaction in an agar gel, instead of doing in a fluid, if you're doing it in a gel, there will be diffusion of antigen and antibody into the gel. That is called as immunodiffusion. So immunodiffusion is a type of precipitation when there is diffusion of antigen and antibody to the gel. And the last type of precipitation reaction is immunoelectrophoresis, where you do electrophoresis to separate the antigens into various components. So then you add the antibody and you get the precipitation. So when you apply electrophoresis to separate antigens into various components, you call it as immunoelectrophoresis. So please learn all these as the types of precipitation reaction. Immunoelectrophoresis, we have two types, counter immunoelectrophoresis and rocket electrophoresis, which you don't have to learn in detail, but you should know what are the different types of immunodiffusion tests. So these are the different types of immunodiffusion tests. There are some named tests. Sometimes you can get MCQs on this. So one is single diffusion in one dimension. You call it as Auden procedure. In this tube, you can see you put antibody in agar gel. This is a gel which contains antibody. Above, you add antigen in solution. What happens is antigen diffuses into the gel, and at the meeting point of equal zone, you get a precipitate at the meeting point. So this is called single diffusion in one dimension. Why it is called single diffusion? Only antigen is diffusing. And why it is called one dimension? Antigen is diffusing only in a single direction. So this procedure is called Odin procedure. And the second one is single diffusion in two dimension. Here what you do is you take a gel containing antibody. So the whole gel is containing antibody. In the middle of the gel, you cut wells where you add the antigens. So this antigen, it will be diffusing like this, not in a one direction, it will be diffusing in a circular or radial fashion into the gel which is containing the antibody. So that is called a single diffusion in two dimensions. So it's not just in a single direction, it's in a circular fashion. So it's called radial immunodiffusion. Now the next one is double diffusion in one dimension. What do you mean by double? Both antigen and antibody are diffusing, but they diffuse only in one direction. You can see a funny name for this procedure, that is Oakley-Fultrop procedure. It's difficult to memorize, but still you can get MCQs, but so you please try to remember. Here there's a tube, here there is antibody in gel, here there is antigen in solution. In between, you're going to put a plain agar. So when there is a plain agar in between, not only this antigen diffuses, antibody also diffuses. That is why it's called double diffusion in one dimension. And you have double diffusion in two dimensions also. That is, both antigen and antibody are diffusing in a radial manner. That's called as Outerloni procedure. Here you can see you put antigen in the center and you put the antibodies in the different wells surrounding it. So there will be diffusion of both the antigens and antibodies in two dimensions. That is why it's called as double diffusion in two dimensions. So when precipitation is asked as a 5-mark question, you have to write the definition, you have to write the types. So in that you have to mention the immunodiffusion methods also. So please remember the most important example of precipitation reaction, that is a flocculation, which we use for the diagnosis of syphilis, VDRL and RPR test. So applications of precipitation reaction, it can be used as a quantitative and also a qualitative test. If you just want to know the presence of antigen or antibody, that's called a qualitative test. If you want to know the quantity of antibody present or antigen present, you call it as a quantitative test. So what are the major applications of precipitation? Ring precipitation test is used for Landsfield's grouping of Streptococcus, which we learn in Streptococcus bacteria. You divide Streptococcus bacteria based on the C carbohydrate antigen present in its cell wall. So based on the different type of C antigens, you put Streptococcus into different groups. So to identify a particular Streptococcus belongs to which group, you take that streptococcal colony and you add that particular antibody and you see whether a precipitation reaction is happening. So that is one application, identification of bacteria. Example is the Lancefield grouping of Streptococcus. Second one is what I told you, the most important example, flocculation test, VDRL in syphilis. It is also used for testing of food adulterants, then presence of toxins, standardized toxins and antitoxins. For all these, we apply precipitation reactions. So these are the applications of precipitation reactions. So the next thing we are going to see is agglutination. So more than precipitation, I'll tell you agglutination is a more important question for exam point of view. So this is what I told you. The only difference between precipitation and agglutination is here the antigen we use is a particulate or insoluble antigen. In precipitation, it was a soluble antigen. Here we use an insoluble antigen. So when an insoluble antigen combines with its antibody in the presence of electrolytes at optimal temperature and pH. So what electrolyte? Sodium chloride. That's we have seen in the previous definition, sodium chloride at optimal temperature and optimal pH. It is forming a bigger complex, bigger than precipitate, because here the antigen itself is insoluble. So it forms a bigger complex in the form of clumping of particles. How will you see agglutination? You can directly visualize it very easily as large clumps of particles, because the antigen antibody complex formed here is bigger. So don't forget, it's a particulate or insoluble antigen that is reacting with its antibody, leading to clumping of the particles because of the formation of complex, antigen antibody complex. So we'll see the types of agglutination reaction. We have mainly four types. One is slide agglutination test, where you do the agglutination in a slide. Tube agglutination test, you perform it in tubes. Heterophile agglutination test and passive agglutination test. We'll see what each of them are. Slide agglutination test, I told you we perform it in slide. The application is, it's used to identify the bacterial strains which are isolated from clinical specimens. So when you grow a bacteria in a lab and you want to know what kind of strain it is, especially it is used in Salmonella, Shigella, Vibrio cholera and all. So you want to know what kind of strain it is. So we have antibodies available for that particular strains. We have reagents commercially available. We take the colony in a slide, you emulsify it with saline and you add the antisera. If you get a visible agglutination or clumping like this, here you can see white clumping is visible in the slide. In the second one, there is no clump. So if you get a positive clumping or a positive agglutination, you take this as positive. So you consider this as that particular strain. For example, for Salmonella, if you put a particular serum against a kind of strain like type 9 Salmonella, you add this anti-serum for that. If you get the visible agglutination, you take it as that particular strain. So that is the use of agglutination, slide agglutination test to identify the bacterial strains. Similarly, this is the best thing which you can use for grouping, blood grouping and cross-matching. So what you do if you want to find out what blood group a particular patient has, you take that patient's RBCs, suppose you add anti-A antibodies. If you get a visible agglutination, that means it is clear that this RBC contains A antigens on the surface. So that is a simple slide agglutination test. So slide agglutination test examples to identify bacterial strains like Salmonella and also for blood grouping and cross-matching. And tube agglutination test is the best test which we use for diagnosis of typhoid fever directly from the patient's serum. You want to know whether the patient's serum contains antibodies against a particular bacteria like Salmonella. We do a test called Vidal test. So the Vidal test is the best example for tube agglutination test. Similarly, for typhus fever, we have a test called Weil-Felix test. So these two are the examples of tube agglutination test. So what you do in a tube agglutination test is, you can see this figure, we add serial dilutions of patient's serum to different tubes. That is, suppose you take the patient's serum, you add 1 ml of serum to 20 ml of saline. So you get a 1/20 dilution. Then you take 1 ml from this tube and you put it to another 20 ml saline. So you get a 1:40 dilution. Like that, if you transfer from this to this like this, you get a doubling dilution, 20, 40, 80, 160 like that. So you have different dilutions of patient serum. So what you can understand, when you increase the dilution, what happened to the concentration of antibody in the tubes? Concentration of antibodies will decrease. Right. So then you're going to add the antigen. So you want to find out whether antibody is present in this serum. So you're going to add the corresponding antigen to each of the tubes. For example, in case of typhoid fever in Vidal, you're going to add Salmonella typhi O antigen to each of the tubes. Now you're going to see you're incubating it at the appropriate temperature, appropriate pH and all, and you're going to see whether antigen antibody reaction has taken place in the form of agglutination. So when the first tube you see, you're seeing a visible agglutination. That means in 1 by 20 dilution, agglutination is positive. Second tube, again it's plus. Third tube, positive. Fourth tube, positive. Fifth tube, 1 is to 320, there is no dilution, there is no agglutination. So that means the highest dilution at which you get a positive agglutination, that is at 1 by 160. So this is the highest dilution where you get a positive agglutination. So that will indicate that is the, in this tube, even the concentration of antibody is very less. In 1 is to 160, that much dilution, the concentration of antibody is very less, still you are getting a positive agglutination. That you take it as the antibody titer. So antibody titer is the highest dilution of serum at which you get a positive agglutination. That is your antibody titer. So in this case, you can take the antibody titer for Salmonella typhi O antibody as 1 is to 160. It's a very good titer that indicates that patient is positive for that particular test. It indicates typhoid fever. So that is tube agglutination test. So what is the advantage of tube agglutination is you can get the exact antibody titer because you can perform serial dilution and you can get a correct antibody titer in tube agglutination test. And please memorize the examples for tube agglutination test, that is Vidal test for typhoid and Wheel Felix test for typhus fever. Now, another example for tube agglutination, the classical example is Coombs test or anti-globulin test. So this was mainly used for detection of anti-RH antibodies in case of fetal incompatibility issues, erythroblastosis fetalis and all. Similarly, it is also used for autoimmune hemolytic anemias. Sometimes patients develop hemolytic anemias due to autoimmune disease. That is, they develop antibodies against their own RBCs. So for diagnosis of such hemolytic anemias also, we use anti-globulin test. There are two types of anti-globulin test or Coombs test. One is direct Coombs test, another is the indirect Coombs test. So we'll see what happened in direct Coombs test and indirect Coombs test. So we'll take an example of autoimmune hemolytic anemia for direct Coombs test, autoimmune hemolytic anemia. So what happened in autoimmune hemolytic anemia? In patients' blood, autoantibodies are produced against their own RBCs. So those antibodies would have been bound to their RBCs. You can see the patient RBCs which are already coated with antibodies. So we take the patient RBCs, if they are already coated with the antibodies, when you add this Coombs reagent is anti-immunoglobulin. If you have an antibody which is produced against another antibody, suppose if you inject human antibodies into rabbit or sheep, what happens? Their body will produce antibodies against this particular, because for them it is foreign. So those antibodies, it's called as antiglobulins or anti-immunoglobulins, where immunoglobulins are produced against an antibody. So you take that antiglobulin against this antibody. Already there is antibody coated to RBCs, you have antiglobulin. So this antiglobulin will bind to these RBCs which are coated with this immunoglobulins antibodies, and you get a positive agglutination. So we have RBCs already coated with antibodies. You add a secondary antibody that is called as antiglobulin, which will cause an agglutination reaction. So that is direct Coombs test. So if this reaction is positive, we assume that the patient's RBCs are coated with antibodies. That means they have autoimmune hemolytic anemia. Similarly, in indirect Coombs test, which is having the most important application in case of screening maternal serum for presence of anti-RH antibodies. Suppose a mother has conceived an RH positive baby. During the first delivery, there will be mixing of blood of mother and fetus baby, leading to production of antibodies in mother's serum. So anti-RH antibodies will be already present in maternal serum. So second time when she again conceives an Rh positive baby, this antibodies can go and react with the fetal RBCs. So that will be dangerous. So in order to know whether the maternal serum is already containing antibodies, what you do is you take the mother's serum. So mother's serum, if suppose it contains anti-Rh antibodies, you add the Rh positive RBCs, they both will bind each other. Then you add the Coombs reagent or the antiglobulin, you get a positive agglutination. So that positive agglutination indicates that this mother's serum is containing anti-Rh antibodies. So this is indirect Coombs test. Why it is indirect Coombs test? Here we are adding the reagent RBCs. In direct Coombs test, you are just taking the patient's RBCs. Here we are adding the reagent RBCs and you are adding the patient's serum. You have to find out whether anti-Rh antibodies are present in the patient's serum. So this is called as indirect Coombs test. So both these tests are done as a tube agglutination method so that you can visualize them very well. Now coming to passive agglutination test is nothing but it's a kind of test where you make a precipitation reaction into an agglutination reaction. You know, in precipitation reaction, the antigens are soluble antigens. So the precipitate means it's very small complex. It's very difficult to visualize. You want to convert it into an agglutination. For that, you have to make the soluble antigen into an insoluble antigen. You have to make the antigen a bigger molecule. So for that, what you commonly do is you take a latex particle and you coat the latex particle with the antigens. So this antigen becomes bigger, becomes insoluble. So when it binds with its antibody, you can see binding of this green color antibodies, you get a visible clumping. So that is passive. So passively you are converting a precipitation reaction to an agglutination reaction. Applications of passive agglutination test are detection of anti-streptolysin O in streptococcal infection, detection of rheumatoid factor for diagnosis of rheumatoid arthritis. So for these kind of tests, we use passive agglutination as a method. And the last type of agglutination reaction, that is heterophile agglutination. I hope you remember heterophile antigens are antigens of different species which have sharing epitopes. So if we are using a heterophile antigen for diagnosis of a particular disease, you call it as a heterophile agglutination test. Best example is Wheel-Felix test. I've told you in Wheel-Felix test, it's for the diagnosis of rickettsial disease. Wheel-Felix test is for diagnosis of rickettsial infection or typhus fever. So what you do here is, we want to find out whether the patient's serum contain antibodies to rickettsia. So we want to know whether the patient's serum contains antibodies to rickettsia. For that, it's very difficult to get rickettsial antigens. What you add to this is you add certain protease strains to this serum. If a reaction occurs, you consider it as positive for rickettsia. Why? Because rickettsial antigens and protease antigens are heterophile antigens. Heterophile antigens, they share epitopes, so antibodies produced against either of them can cross-react. So we are using reagent, we use an antigen of a different species in order to find out whether antibodies are produced against another species. So that is the application of heterophile agglutination test. And another example is, other than Wheel-Felix, is Paul Bunnell test. Paul Bunnell test is for diagnosis of infectious mononucleosis, where you use sheep RBC as the reagent. So if in infectious mononucleosis, the patient's serum contains antibodies which can cross-react with sheep RBCs. So we use sheep RBC as a reagent. So both these are examples for heterophile agglutination test. So with that, we have finished precipitation and agglutination reaction. Agglutination is more important. Please learn all the types of agglutination with examples and don't forget the definition, that is, we are using an insoluble antigen. So that is about the precipitation and agglutination reactions. Thank you.